Hello there, and welcome to my channel, where the mysteries of our world unfold gently like a soft blanket on a quiet evening. I'm so glad you found your way here tonight. Whether you've had a long day wrestling with deadlines, a restless mind that won't quite settle, or you simply love drifting off to the gentle hum of science, this video was made just for you. Tonight, we're embarking on a peaceful journey through one of the most fascinating and surprisingly elegant dances in nature. We'll be exploring how tiny, invisible entities called viruses change and adapt over time, and how these microscopic transformations can ripple outward to touch the lives of millions. It's the quiet science behind pandemics, told in the most soothing way possible. But before we begin our gentle exploration, I'd love to know where in the world you're listening from right now, and what time it is in your corner of the universe. Are you tucked under warm covers with the city lights twinkling outside your window? Or perhaps curled up somewhere peaceful and dark with a cup of something warm nearby? There's something rather beautiful about knowing that we're all scattered across different time zones, each arriving at this moment from our own unique slice of the day. If you find these sleepy scientific journeys calming or intriguing, I'd be ever so grateful if you gave the video a gentle like and perhaps tapped that subscribe button. It helps other curious souls find their way to this tranquil corner of learning, and it means I can keep crafting more of these soft explorations for you. Now, I do hope this isn't so captivating that it accidentally keeps you awake, though if it does, at least you'll drift off knowing a little more about how our microscopic world works. Not such a terrible trade, really. So settle in, let your breathing slow, and let's take a wonderfully unhurried wander through the hidden world of viral evolution and the gentle science that helps us understand how pandemics unfold. Viruses are perhaps the most misunderstood characters in the story of life on Earth. They're neither fully alive nor completely lifeless, existing in that curious space between being and non-being. Think of them as nature's most minimalist travelers, carrying only the bare essentials for their journey through the world. They have no cells of their own, no way to reproduce without a host, no metabolism to speak of. And yet, in their elegant simplicity, they've mastered one of life's most fundamental skills, the ability to change. When we think about evolution, we often picture grand sweeping changes happening over millions of years. Dinosaurs growing feathers, fish developing legs, flowers learning to bloom in brilliant colors. But viral evolution happens on a completely different timescale, one that's both intimate and immediate. While you sleep peacefully through a single night, a virus might go through dozens of generations, each one a tiny experiment in survival. Imagine, if you will, a virus as a simple recipe written in the universal language of genetic code. This recipe is remarkably short, sometimes containing instructions for just a handful of proteins. But here's where it gets interesting. Every time this recipe is copied, there's a chance for a small mistake to creep in. A letter might be swapped for another, a line might be skipped, or sometimes a whole new ingredient gets added to the mix. Most of these mistakes are harmless, like changing the font on a document without altering the meaning. Some are harmful to the virus itself, creating versions that simply can't survive. But occasionally, very rarely, one of these tiny copying errors creates something new, something that works just a little bit better than the original. And when that happens, natural selection steps in like a gentle editor, quietly favoring the improved version. This process is called mutation, and it's happening constantly, silently all around us. Every time a virus reproduces inside a host cell, it's rolling the dice on change. The vast majority of these rolls come up empty, but the few that succeed become the foundation for viral evolution. It's a numbers game played out in the microscopic realm, where billions of attempts yield just a handful of meaningful improvements. But viruses have another trick up their sleeve, one that makes them particularly clever at adapting to new challenges. When two different viruses infect the same cell at the same time, they can engage in a process called recombination. Think of it as a microscopic dance where genetic material gets shuffled and shared, like dancers swapping partners mid-song. This allows viruses to combine successful mutations from different lineages, creating hybrid versions that might be better adapted to their environment. The environment, of course, is us, and other animals, and plants, and bacteria. Viruses are the ultimate opportunists, each one specialized for a particular type of host. Some prefer birds, others mammals. Some target the respiratory system, others the digestive tract. Each virus has evolved to fit its chosen niche like a key fits a lock, and that specialization shapes how it evolves. When a virus that normally infects animals suddenly finds its way into humans, something remarkable and sometimes concerning happens. This process is called zoonotic transmission, and it's the origin story of many pandemic diseases. The virus finds itself in an entirely new environment, facing immune systems it's never encountered before, trying to navigate cellular machinery that's subtly different from what it knows. Most of the time, these cross-species jumps fail. The virus simply can't adapt quickly enough to its new host, and it dies out before it can establish itself. But sometimes, through a combination of mutations and sheer chance, a virus manages to find its footing in human cells. And when that happens, the real evolutionary race begins. Picture a virus that has just made the leap from animals to humans. It's like a traveller arriving in a foreign country where they don't speak the language and don't understand the customs. At first, it might struggle to reproduce effectively, to spread from person to person, to avoid detection by the immune system. But with each generation, with each round of mutation and selection, it gets a little better at being a human virus. This adaptation isn't random. It follows predictable patterns that scientists have learned to recognize. A virus might evolve to bind more tightly to human cell receptors, the molecular doorways it uses to gain entry. It might develop ways to evade antibodies, the immune system's security guards. It might become more or less severe in its effects, depending on what strategy helps it spread most effectively. The relationship between virulence and transmission is particularly fascinating. You might think that a virus would always evolve to become more deadly, but that's not necessarily the case. A virus that kills its host too quickly doesn't have much opportunity to spread to new hosts. So there's often an evolutionary pressure toward a middle ground, where the virus is successful enough to reproduce and spread, but not so aggressive that it eliminates its own opportunities for transmission. This is why many pandemic diseases seem to follow a predictable pattern. They often begin with high mortality rates when they first jump to humans, but over time, they may evolve to become less severe. It's not because the virus is becoming kinder, but because the variants that allow for better transmission often happen to be less lethal to the host. Of course, this isn't a universal rule. Some viruses find their optimal strategy in rapid reproduction and quick transmission, regardless of the consequences to the host. Others develop the ability to persist in the host for long periods, spreading slowly but steadily. The evolutionary pressures are different for each virus, shaped by how they transmit, what cells they target, and how the host's immune system responds. The human immune system, for its part, is engaged in its own evolutionary dance with viruses. Over generations, populations that have been exposed to particular diseases develop genetic changes that provide some protection. This is why certain infectious diseases that devastated naive populations had less severe effects on groups that had been exposed to them for centuries. But individual immune systems are also remarkably adaptable. When you recover from a viral infection, your immune system retains a memory of that virus, allowing for a faster and more effective response if you encounter it again. This immunological memory is the foundation of vaccination, where we can train the immune system to recognize and fight a virus without having to experience the full disease. Yet viruses have evolved counter-strategies, even to this remarkable defense system. Some viruses mutate so rapidly that they can stay one step ahead of immune memory, constantly changing their appearance just enough to avoid recognition. Others target the immune system directly, weakening the very defenses they're trying to evade. The speed of viral evolution presents both challenges and opportunities for human health. On one hand, it means that viruses can rapidly develop resistance to treatments or vaccines, requiring us to constantly update our medical interventions. On the other hand, it provides us with a window into the evolutionary process itself, allowing us to study adaptation and selection in real time. Modern molecular techniques have given us unprecedented insight into viral evolution. We can now sequence the genetic code of viruses quickly and cheaply, tracking how they change as they spread through populations. This has revealed the intricate family trees of viral lineages, showing us how different variants are related to each other and how they've adapted to different environments. These genetic fingerprints tell stories of viral journeys across continents, of adaptive changes that occurred in response to specific challenges, of the constant arms race between viruses and their hosts. They've helped us understand not just where pandemic viruses come from, but how they're likely to evolve in the future. The patterns we see in viral evolution are surprisingly consistent across different types of viruses. RNA viruses, which include many respiratory pathogens, tend to evolve more rapidly than DNA viruses because their copying machinery is less accurate. This higher mutation rate gives them more opportunities for adaptive change, but it also means they're more likely to accumulate harmful mutations. DNA viruses, by contrast, tend to evolve more slowly and steadily. Their copying machinery has built-in proofreading mechanisms that reduce the mutation rate, leading to more stable genetic sequences. This doesn't make them less dangerous, just different in their evolutionary strategies. Some viruses have evolved mechanisms that actually increase their mutation rates in specific parts of their genome, essentially turning up the evolutionary pressure in areas where adaptation is most beneficial. Others have developed ways to recombine more frequently, increasing the speed at which they can acquire new beneficial traits. The environment in which a virus evolves also shapes its trajectory. A virus spreading through a highly connected urban population faces different selective pressures than one circulating in isolated rural communities. Population density, travel patterns, healthcare interventions, and social behaviors all influence which viral variants are most successful. As we lie here contemplating these microscopic dramas playing out all around us, it's worth remembering that viral evolution isn't inherently good or bad. It simply is. These tiny entities are following the same fundamental rules that govern all life on Earth. Survive, reproduce, and pass on your genetic material to the next generation. The fact that this sometimes creates challenges for human health is largely incidental from the virus's perspective. Understanding viral evolution helps us appreciate both the elegance and the inevitability of pandemic diseases. They're not random catastrophes that strike without warning, but predictable consequences of the way life works on our planet. By studying these patterns, we can better prepare for future challenges and develop more effective strategies for protecting human health. The story of how viruses spread from one host to another is like watching a slow-motion ballet performed on a global stage. Each pandemic begins with what epidemiologists call patient zero, though in reality, there's rarely just one patient. More often, it's a quiet cascade of infections, spreading through communities like ripples on a still pond, mostly invisible until the waves grow large enough to notice. Imagine a virus that has successfully adapted to human hosts, having made that crucial leap from its animal reservoir. Now it faces a new challenge: how to move efficiently from person to person in a world of 7 billion potential hosts. This is where the mathematics of viral spread becomes both beautiful and sobering, following patterns that have remained remarkably consistent throughout human history. The basic reproduction number, which scientists call R0, is perhaps the most elegant concept in all of epidemiology. It represents the average number of people that one infected person will infect in a completely susceptible population. If R0 is less than 1, the outbreak fizzles out like a dying ember. If it's greater than 1, the virus spreads exponentially, each generation infecting more people than the last. But R0 isn't fixed in stone. It's shaped by the biology of the virus the behavior of the hosts, and the environment in which transmission occurs. A virus that spreads through respiratory droplets will have a different R0 in a crowded indoor space than in an open field. Social distancing measures, mask wearing, and vaccination all work by reducing the effective reproduction number, making it harder for each infected person to pass the virus to others. The early days of a pandemic often follow a deceptively gentle curve. A few cases here, a small cluster there, numbers that seem manageable and contained. This is the exponential phase in its infancy, when each infected person is creating multiple new infections, but the absolute numbers are still small. It's like compound interest in a savings account, modest at first but powerful over time. Then comes the moment when exponential growth reveals its true nature. Cases double, then double again and again. What seemed like a local problem suddenly becomes regional, then national, then global. This is when public health officials start using words like outbreak, epidemic, and eventually pandemic. The transition from one to the next isn't always clear-cut, but it reflects the virus's success in establishing sustainable chains of transmission across multiple populations. Different viruses have evolved different strategies for achieving this spread. Some, like measles, are incredibly contagious but create lifelong immunity in survivors, eventually running out of susceptible hosts. Others, like influenza, constantly change their surface proteins, allowing them to reinfect the same populations year after year. Still others find ways to persist in hosts for long periods, spreading slowly but steadily without triggering strong immune responses. The seasonality of many viral diseases reflects deep evolutionary adaptations to environmental conditions. Influenza thrives in the dry, cold air of winter, when people spend more time indoors and the virus particles remain stable longer in the air. Some enteric viruses prefer the warmer months, when increased travel and outdoor activities create more opportunities for transmission. These patterns have held true for centuries, creating predictable rhythms in the ebb and flow of infectious disease. Modern transportation has fundamentally altered the dynamics of pandemic spread. A virus that once would have taken months or years to cross continents can now circle the globe in a matter of days, carried by unwitting passengers on international flights. Airport hubs become crucial nodes in the global network of transmission, and the patterns of airline routes can predict how quickly a new pathogen will spread to different regions. Yet even in our interconnected world, geography still matters. Mountain ranges, deserts, and bodies of water can serve as natural barriers to transmission. More importantly, economic and social factors create invisible boundaries that influence how viruses spread through populations. Rural communities might remain isolated from urban outbreaks for extended periods, while densely populated cities with extensive public transportation systems can see rapid amplification of transmission. The concept of herd immunity emerges naturally from the mathematics of viral spread. As more people in a population become immune, either through infection or vaccination, the virus finds it increasingly difficult to find susceptible hosts. The threshold for herd immunity varies depending on the contagiousness of the virus, but once reached, it provides indirect protection even to those who remain susceptible. However, achieving herd immunity through natural infection comes at a tremendous cost in human suffering. The virus must spread through a large portion of the population, causing illness, death, and long-term health consequences along the way. This is why vaccination is such a powerful tool, allowing populations to develop immunity without experiencing the full burden of disease. The heterogeneity of human populations adds layers of complexity to pandemic dynamics. Some people are super spreaders, infecting far more contacts than average due to biological factors, behavioral patterns, or simply being in the wrong place at the wrong time. Others seem naturally resistant to infection, or less likely to transmit the virus once infected. These variations create hot spots and cold spots in the landscape of transmission, making the spread of disease uneven and sometimes unpredictable. Age plays a crucial role in many pandemic diseases, with different viruses showing distinct preferences for certain age groups. Children might be more susceptible to some viruses while being largely spared by others. Elderly populations often bear a disproportionate burden of severe disease, reflecting both age-related changes in immune function and a lifetime of exposure to related pathogens. The social determinants of health become starkly visible during pandemics. Crowded housing conditions, jobs that require physical presence, limited access to healthcare, and underlying health conditions all influence who gets infected and who suffers the most severe consequences. Pandemics don't affect all populations equally, and these disparities reflect broader patterns of social and economic inequality. Public health interventions work by disrupting the chains of transmission that viruses depend upon. Contact tracing attempts to identify and isolate infected individuals before they can spread the virus further. Quarantine measures separate potentially infected people from the general population. Social distancing reduces the number of contacts each person has, lowering the average number of transmission opportunities. The timing of these interventions matters enormously. Early in an outbreak, when case numbers are still small, aggressive contact tracing and isolation can sometimes contain the spread entirely. But once community transmission becomes widespread, the focus shifts to broader population-level measures designed to slow the spread and prevent healthcare systems from becoming overwhelmed. Vaccines represent humanity's most sophisticated response to pandemic viruses. By training the immune system to recognize and respond to specific pathogens, vaccines can prevent infection entirely or reduce the severity of disease. The development of effective vaccines requires a deep understanding of viral biology, immune responses, and the specific features that make each virus unique. The race to develop vaccines during a pandemic is a remarkable example of scientific collaboration and innovation under pressure. Traditional vaccine development takes years or even decades, but modern techniques and unprecedented global cooperation have dramatically shortened these timelines while maintaining safety standards. The platforms developed for one pandemic can often be rapidly adapted for future threats. Antiviral medications offer another layer of defense, particularly for viruses where vaccines are not available or not fully effective. Unlike antibiotics, which work against a broad range of bacterial infections, antiviral drugs are typically highly specific to particular viruses or viral families. This specificity makes them powerful tools when available, but also means that each new pandemic virus may require the development of new therapeutic approaches. The global nature of modern pandemics requires unprecedented levels of international cooperation. Viruses don't respect national borders, and effective pandemic response requires sharing of information, resources, and expertise across political boundaries. Early warning systems, global surveillance networks, and international health regulations all play crucial roles in detecting and responding to emerging threats. Yet despite all our scientific knowledge and technological capabilities, predicting the exact course of a pandemic remains challenging. The complex interactions between viral evolution, human behavior, public health measures, and social factors create a system that is inherently difficult to forecast with precision. Models can provide valuable insights and help guide decision-making, but they are always approximations of a reality that is far more complex. The psychological and social impacts of pandemics extend far beyond the direct health effects of the virus itself. Fear, uncertainty, and disruption to normal routines can have profound effects on mental health and social cohesion. The measures needed to control viral spread, such as isolation and social distancing, can themselves create new challenges for human well-being. Economic disruptions during pandemics create cascading effects throughout society. Industries dependent on human interaction, such as hospitality and entertainment, may face existential threats. Supply chains that seemed robust in normal times reveal unexpected vulnerabilities. The economic costs of pandemic response must be weighed against the health benefits, creating difficult trade-offs for policymakers. Education systems face particular challenges during pandemics, especially when children and young adults are significant vectors of transmission. School closures can effectively reduce viral spread, but come at enormous costs to student learning and development. The shift to remote learning during recent pandemics has highlighted both the potential and the limitations of technology-based education. The information landscape during pandemics has become increasingly complex in our digital age. Social media platforms can rapidly spread both accurate information and dangerous misinformation about viral spread, treatments, and preventive measures. The challenge of maintaining public trust while communicating uncertainty and changing recommendations has become a central aspect of pandemic response. As we drift peacefully through these thoughts about viral spread and pandemic dynamics, it's worth reflecting on the remarkable resilience of human societies. Throughout history, we faced countless infectious disease challenges, and while each pandemic brings tragedy and disruption, we've consistently found ways to adapt, learn, and emerge stronger. The scientific understanding we've developed, the public health infrastructure we've built, and the international cooperation we've fostered all represent humanity's collective response to these microbial challenges. The next pandemic will inevitably come, as surely as the seasons change and the tides ebb and flow. But our understanding of viral evolution and pandemic dynamics continues to deepen. Our tools for detection and response continue to improve, and our capacity for global cooperation continues to evolve. In the quiet spaces between one pandemic and the next, scientists work tirelessly to better understand these invisible threats, developing new vaccines, treatments, and strategies that will serve us well when the next challenge arrives. The story of how we detect and respond to emerging viral threats is like watching a global network of sentries standing guard at the edges of the known world. These modern sentinels aren't stationed on castle walls or mountain peaks, but in laboratories, hospitals, and research stations scattered across every continent, quietly monitoring the microscopic realm for signs of change. Disease surveillance has evolved from the ancient practice of quarantine ships in medieval harbours to a sophisticated web of molecular detection systems that can identify new viral variants within days of their emergence. At its heart, surveillance relies on the simple principle that unusual patterns in human and animal health often herald the arrival of something new. A cluster of unexplained respiratory illness in a remote village, an uptick in severe pneumonia cases at a city hospital, or unusual mortality in wild bird populations can all serve as early warning signals. The World Health Organization coordinates much of this global watching, but the real work happens at the local level. Rural doctors noticing that their patients' symptoms don't quite match familiar patterns, veterinarians observing strange illnesses in livestock, and laboratory technicians seeing unusual results in routine tests all contribute to this vast intelligence network. Their observations flow upward through national health systems and international reporting mechanisms, creating a kind of early warning system for humanity. Modern molecular techniques have revolutionized how quickly we can identify and characterize new viral threats. What once required weeks of painstaking laboratory work can now be accomplished in hours. Genetic sequencing technology allows scientists to read the complete genetic code of a virus from a single patient sample, revealing not only what type of virus it is, but also how it relates to known strains and what mutations it might have acquired. This rapid genetic analysis has become crucial for understanding how viruses are spreading and evolving in real time. Scientists can now track the movement of specific viral lineages across continents, identify which variants are becoming more common, and spot concerning mutations as they emerge. It's like having a microscopic GPS system that can trace the journey of individual viral particles as they move through human populations. The concept of genomic epidemiology has emerged from this technological revolution. Instead of relying solely on traditional epidemiological methods like interviewing patients about their contacts and travel history, scientists can now use genetic similarities between viral samples to reconstruct transmission chains. If two patients have viruses with nearly identical genetic sequences, they were likely infected by the same source or by people very close in the transmission chain. This genetic sleuthing has revealed surprising insights about how viruses spread. Sometimes patients who seem completely unconnected epidemiologically turn out to share viral strains that are nearly identical, suggesting hidden transmission pathways. Other times, patients who were in close contact carry viruses that are genetically distinct, indicating they were infected from different sources entirely. The speed of modern communication means that information about new viral threats can circle the globe even faster than the viruses themselves. Within hours of identifying a concerning new pathogen, scientists can share genetic sequences, clinical observations, and epidemiological data with colleagues worldwide. This rapid information sharing allows other countries to begin preparing their detection systems and response plans before the virus even arrives on their shores. Yet, for all our technological sophistication, the human element remains central to pandemic preparedness. The rural physician who recognizes that something doesn't seem quite right, the laboratory worker who notices an unusual pattern in test results, the public health official who connects seemingly unrelated reports, these individual observations and insights are still the foundation upon which our entire surveillance system rests. The challenge of detecting new pandemic threats is complicated by the sheer diversity of places where dangerous viruses might emerge. Wildlife markets in Southeast Asia, poultry farms in Eastern Europe, research laboratories in major cities, and remote forest communities where humans increasingly interact with wild animals all represent potential emergent sites. Each setting requires different surveillance strategies and presents unique logistical challenges. Animal surveillance has become increasingly important as we've recognized that most pandemic viruses originate in non-human species before making the jump to humans. Monitoring wild bird populations for influenza variants, testing bats for coronaviruses, and screening livestock for unusual respiratory infections all contribute to our understanding of the viral threats circulating in animal reservoirs. The One Health approach recognizes that human, animal, and environmental health are interconnected, and that effective pandemic prevention requires monitoring all three domains simultaneously. A new virus detected in wild animals might pose no immediate threat to humans, but continued surveillance could reveal when it begins acquiring mutations that make human infection more likely. Climate change is altering the landscape of viral emergence in ways we're only beginning to understand. As temperatures rise and precipitation patterns shift, the geographic ranges of animal species are changing, bringing new combinations of hosts and pathogens into contact. Melting permafrost may release ancient viruses that have been frozen for thousands of years, while changing weather patterns affect the breeding cycles and migration routes of animals that serve as viral reservoirs. Urbanization and deforestation create new interfaces between human populations and wildlife, increasing opportunities for cross-species viral transmission. As cities expand into previously wild areas and agricultural practices intensify, humans come into closer and more frequent contact with animal species and their associated pathogens. The global trade in wildlife, both legal and illegal, creates pathways for viruses to move rapidly across continents. An exotic pet infected with a novel virus, bushmeat transported from remote forests to urban markets, or live animals traded for traditional medicine, can all serve as vehicles for introducing new pathogens to susceptible human populations. Laboratory security has become a critical concern as research facilities around the world study increasingly dangerous pathogens. While these laboratories play essential roles in developing vaccines and treatments, they also represent potential sources of accidental release. Stringent safety protocols, regular inspections, and international oversight help minimize these risks. but the possibility of laboratory accidents remains a concern for pandemic preparedness. The concept of dual-use research has emerged as scientists grapple with studies that could enhance our understanding of viral threats while simultaneously providing information that could be misused. Research that makes viruses more transmissible or virulent, even when conducted for legitimate scientific purposes, raises complex questions about the balance between advancing knowledge and maintaining security. International cooperation in pandemic preparedness faces numerous challenges, from political tensions between nations to disparities in scientific capabilities and resources. Sharing information about emerging threats requires trust and transparency, while developing coordinated response plans requires sustained commitment and investment from multiple countries with different priorities and capabilities. The COVID-19 pandemic revealed both strengths and weaknesses in our global preparedness systems. The rapid development and sharing of viral genetic sequences demonstrated the power of international scientific collaboration, while delays in information sharing and coordination highlighted areas where improvements are needed. Vaccine development platforms have evolved to become more flexible and rapid in their response to new threats. Instead of starting from scratch with each new pandemic virus, scientists can now adapt existing vaccine technologies to target novel pathogens. This has dramatically reduced the time needed to develop effective vaccines, though challenges remain in manufacturing and distributing them globally. Stockpiling strategies have become more sophisticated, with countries maintaining reserves not just of vaccines and medications, but also of raw materials, manufacturing equipment, and specialized supplies needed for pandemic response. The challenge lies in balancing the costs of maintaining these stockpiles with the uncertainty about when they might be needed and which specific threats they should address. Communication strategies for pandemic preparedness must navigate the complex landscape of public trust, media coverage, and social media amplification. Preparing populations for potential threats without causing panic, maintaining credibility while acknowledging uncertainty, and countering misinformation while respecting diverse viewpoints all present ongoing challenges for public health officials. The economic dimensions of pandemic preparedness require long-term thinking and investment in areas that may not show immediate returns. Building surveillance systems, training personnel, maintaining stockpiles, and conducting research all require sustained funding during periods when pandemic threats may seem distant and abstract. Community engagement has emerged as a crucial component of effective pandemic preparedness. Local communities are often the first to detect unusual health patterns and the last line of defense in implementing control measures. Building trust, providing education, and ensuring that preparedness plans are culturally appropriate and practically feasible all contribute to more effective responses. The role of technology in pandemic preparedness continues to expand, from artificial intelligence systems that can detect unusual patterns in health data to mobile applications that can facilitate contact tracing and symptom monitoring. However, the effectiveness of these technological solutions depends heavily on public acceptance and participation. Privacy concerns have become increasingly important as digital tools for pandemic surveillance and response become more sophisticated. Balancing the public health benefits of data collection and sharing with individual privacy rights requires careful consideration of legal, ethical, and practical factors. The psychological preparedness of populations for pandemic threats involves helping people understand risks without becoming overwhelmed by anxiety, providing practical guidance for protective behaviors, and building resilience for the social and economic disruptions that pandemics can cause. Healthcare system preparedness extends beyond simply having enough hospital beds and ventilators to include surge capacity planning, cross-training of personnel, supply chain resilience, and coordination between different levels of care. The goal is to maintain essential services while accommodating the additional demands of pandemic response. As we rest quietly in contemplation of these vast preparedness efforts unfolding around us, It's remarkable to consider how much of this work happens in the background of our daily lives. Scientists in laboratories around the world are sequencing viral genomes. Epidemiologists are analyzing disease patterns, and public health officials are updating response plans, all working to protect populations from threats that may never materialize. This constant vigilance represents one of humanity's most ambitious collective endeavors, a global commitment to protecting our species from microscopic threats that respect no borders and acknowledge no sovereignty. The systems we've built for pandemic preparedness reflect our growing understanding that infectious diseases are fundamentally a shared challenge, requiring shared solutions and sustained cooperation across all boundaries that typically divide us. The next viral threat may emerge tomorrow or may remain dormant for decades, but our preparation continues regardless. In research laboratories and surveillance systems, in international meetings and local planning committees, in the quiet dedication of scientists and public health workers around the world, humanity maintains its watch against the invisible dangers that have shaped our history and will undoubtedly influence our future. The relationship between humans and viruses is far more nuanced than the simple narrative of us versus them that often dominates our thinking. In reality, we exist in a delicate ecological balance with these microscopic entities, a relationship that has been evolving for as long as life has existed on Earth. To understand pandemics, we must first appreciate that viruses are not invaders from some distant realm, but intimate partners in the dance of evolution that has shaped all living things. Consider for a moment that viruses may have played a crucial role in the very origins of life itself. Some scientists theorize that these simple genetic packages were among the earliest replicating entities on our planet, existing in primordial seas billions of years ago. They may have been the first to discover the fundamental trick of using one molecule to make copies of itself, a discovery that would eventually give rise to all the complexity we see in the living world today. Even now, viruses continue to drive evolution in ways both subtle and profound. They serve as vectors for horizontal gene transfer, carrying genetic material between different species and even between different kingdoms of life. A virus that infects a bacterium might pick up a useful gene and later transfer it to a plant or an animal, facilitating the spread of beneficial traits across the tree of life in ways that would be impossible through normal reproduction alone. Our own genomes tell the story of this ancient partnership. Roughly 8% of human DNA consists of sequences derived from ancient viral infections, genetic fossils of viruses that once infected our distant ancestors and became permanently integrated into our hereditary material. These endogenous retroviruses, as they're called, are now part of what makes us human, and some of them appear to play important roles in normal development and physiology. The placenta that nourishes developing babies contains proteins that were originally derived from viral genes, repurposed over millions of years of evolution to serve essential functions in mammalian reproduction. In a very real sense, we couldn't exist without the genetic contributions of ancient viruses that infected our evolutionary ancestors long before humans walked the earth. This intimate relationship extends to our microbiome, the vast community of bacteria, fungi, and other microorganisms that live in and on our bodies. Many of these microbial partners harbor their own viruses, creating complex, multi-layered ecosystems where viral, bacterial, and human evolution all influence each other. Changes in our viral load can affect our bacterial communities, which in turn influence our immune system, our metabolism, and even our mood and behavior. The human immune system itself bears the marks of its long coevolution with viruses. Many of our most sophisticated immune mechanisms evolved specifically to deal with viral threats. The ability to present viral proteins on cell surfaces to alert immune cells, the development of specialized killer cells that can recognize and destroy infected cells, the complex antibody systems that can remember and respond to previously encountered viruses. All of these represent evolutionary innovations driven by the constant pressure of viral infection. Yet viruses have not remained passive in this evolutionary arms race. They have developed an equally impressive array of strategies to evade, subvert, and manipulate our immune responses. Some viruses produce proteins that mimic our own immune signaling molecules, sending false messages that confuse our defenses. Others target the immune system directly, destroying the very cells that are meant to protect us from infection. This ongoing evolutionary dialogue between hosts and viruses creates what biologists call the Red Queen hypothesis, named after the character in Lewis Carroll's Through the Looking Glass, who had to keep running just to stay in the same place. Both viruses and their hosts must constantly evolve new strategies and countermeasures just to maintain their position in the struggle for survival. The speed of this evolutionary race varies dramatically depending on the type of virus and host involved. RNA viruses, with their high mutation rates and short generation times, can evolve new variants within days or weeks. Their hosts, with longer generation times and more stable genomes, evolve much more slowly. This creates an asymmetry where viruses can often stay one step ahead of host defenses, at least in the short term. But hosts have their own advantages in this evolutionary game. Our immune systems can adapt within an individual lifetime, learning to recognize and respond to new viral threats without waiting for genetic changes to accumulate over generations. This immunological memory allows us to develop resistance to viruses we've never encountered before, simply by being exposed to related strains or carefully designed vaccines. The concept of antigenic drift illustrates how this evolutionary dance plays out over time. Influenza viruses gradually accumulate small mutations in their surface proteins, slowly changing their appearance to the immune system. Each year's flu vaccine must be reformulated to account for these changes, as last year's version becomes less effective against this year's evolved variants. Occasionally, influenza undergoes a more dramatic change called antigenic shift, where genetic material from different viral strains recombines to create a hybrid with substantially different properties. These shift events can produce pandemic strains that spread rapidly through populations with little existing immunity, as happened with the 1918 Spanish flu, the 1957 Asian flu, and the 2009 H1N1 pandemic. The 1918 influenza pandemic offers a particularly instructive example of how viral evolution, host immunity, and social factors interact during pandemic events. The virus that caused this pandemic was an H1N1 strain that had recently acquired the ability to transmit efficiently between humans. What made it so deadly was not just its biological properties, but also the social conditions of the time. World War I had created perfect conditions for viral spread, with millions of young adults crowded into military camps, transported in cramped ships and trains, and subjected to the stress and malnutrition that weaken immune systems. The virus spread along military supply lines and troop movements, reaching remote corners of the globe with unprecedented speed. But the 1918 pandemic also demonstrated the power of social interventions to modify viral spread. Cities that implemented early and aggressive social distancing measures, such as school closures and bans on public gatherings, experienced lower mortality rates than those that delayed or avoided such measures. The virus itself was the same, but human behavior shaped how severely different communities were affected. The aftermath of the 1918 pandemic reveals another important aspect of human viral evolution. The H1N1 strain that caused the pandemic didn't disappear after 1918, but continued circulating in human populations for decades, gradually evolving to become less severe as both the virus and human immune systems adapted to each other. By the 1952nd, seasonal H1N1 influenza was a manageable annual occurrence rather than a catastrophic pandemic threat. This pattern of initial pandemic severity, followed by gradual attenuation, has been observed with other pandemic viruses as well. The 2009 H1N1 pandemic was relatively mild compared to historical pandemics, partly because it was caused by a virus that was genetically similar to strains that had circulated decades earlier, meaning that older adults had some residual immunity from childhood exposures. The role of children in viral evolution and transmission presents another fascinating aspect of human-virus coevolution. Children are often described as mixing vessels for respiratory viruses because they have frequent close contact with each other and haven't yet developed immunity to as many viral strains as adults. This makes them important drivers of viral evolution and transmission, but also means that their immune systems are constantly being educated about new viral threats. Childhood viral infections often produce lifelong immunity that shapes how individuals respond to related viruses encountered later in life. The phenomenon of original antigenic sin describes how our first exposure to a particular type of virus can bias our immune responses to related viruses encountered decades later, sometimes in ways that are protective and sometimes in ways that are harmful. The hygiene hypothesis suggests that reduced exposure to viruses and other microorganisms during childhood might actually weaken our immune systems, making us more susceptible to allergies, autoimmune diseases, and severe infections later in life. This counterintuitive idea highlights how our coevolution with viruses has shaped not just our immune systems, but our entire approach to child rearing and public health. Modern medical interventions have dramatically altered the landscape of human-virus coevolution. Vaccines allow us to develop immunity without experiencing the full consequences of infection, while antiviral medications can reduce viral replication and transmission. These interventions represent unprecedented tools for influencing the evolutionary relationship between humans and viruses. However, these same interventions also create new selective pressures that can drive viral evolution in unexpected directions. Vaccines that don't completely prevent infection might favor viral variants that can partially evade vaccine immunity. Antiviral medications can select for drug-resistant variants. The challenge is to design interventions that provide protection while minimizing the risk of driving harmful viral evolution. The concept of vaccine-induced herd immunity represents one of humanity's most successful strategies for managing viral threats. By achieving high vaccination coverage in populations, we can interrupt viral transmission chains and protect even those individuals who cannot be vaccinated themselves. This collective protection has allowed us to eliminate some viral diseases entirely, such as smallpox, and bring others to the brink of elimination, such as polio. Yet, achieving and maintaining herd immunity requires sustained effort and vigilance. Vaccination rates must be maintained at high levels, and surveillance systems must monitor for breakthrough infections and vaccine escape variants. The recent resurgence of measles in some populations with declining vaccination rates demonstrates how quickly vaccine-preventable diseases can return when herd immunity is compromised. The social and cultural dimensions of human-virus relationships add another layer of complexity to pandemic dynamics. Different societies have developed different cultural practices around illness, hygiene, and social contact, all of which influence how viruses spread through populations. Traditional healing practices, religious gatherings, funeral customs, and family structures all play roles in shaping transmission patterns. Globalization has fundamentally altered these traditional patterns, creating new pathways for viral spread while disrupting some of the cultural practices that historically provided protection. International travel, global trade, urbanization, and changing social structures have created a world where viruses can spread faster and farther than ever before, but where traditional community-based responses may be less effective. The psychological aspects of pandemic response reflect deep-seated human instincts about disease and social cooperation. Fear of contagion is one of our most ancient and powerful emotions, shaped by millions of years of evolution in which infectious diseases were major sources of mortality. This fear can motivate protective behaviors, but it can also lead to stigmatization, social fragmentation, and irrational responses that may actually facilitate viral spread. Trust in institutions, scientific expertise, and community leaders plays a crucial role in pandemic response. Effective public health measures require widespread cooperation and compliance, which depend on social cohesion and shared understanding of threats and appropriate responses. When trust breaks down, even well-designed interventions may fail to achieve their intended effects. The information environment during pandemics has become increasingly complex in our digital age. Social media platforms can rapidly amplify both accurate information and dangerous misinformation about viral threats and interventions. The challenge of maintaining public understanding and cooperation while acknowledging scientific uncertainty has become a central aspect of pandemic management. Economic factors interweave with biological and social factors to shape pandemic outcomes. Poverty, inequality, and social marginalization create conditions that facilitate viral transmission while limiting access to protective measures and healthcare. The economic disruptions caused by pandemic control measures can themselves have health consequences, creating difficult trade-offs between different types of harm. As we contemplate these intricate webs of interaction between humans and viruses, it becomes clear that pandemics are not simply biological events, but complex social phenomena that reflect the full spectrum of human organization and behavior. Understanding and preparing for future pandemics requires not just biological and medical knowledge, but insights from sociology, psychology, economics, anthropology, and many other fields. The story of human-virus coevolution is still being written, with new chapters added every day as viruses continue to evolve and human societies continue to change. Our growing scientific understanding, technological capabilities, and global cooperation provide unprecedented tools for managing viral threats, but they also create new challenges and uncertainties that we're still learning to navigate. Perhaps most remarkably, this ancient relationship between humans and viruses continues to shape us in ways both visible and hidden. Every breath we take, every interaction we have, every decision we make about health and social behavior contributes to the ongoing evolutionary dance between our species and the microscopic entities that have been our constant companions throughout the long journey of life on Earth. In understanding this relationship, we begin to appreciate not just the challenges that viruses present, but also the profound ways they have contributed to making us who we are. The future of pandemic preparedness lies not just in our laboratories and hospitals, but in the quiet revolution happening in how we understand and predict viral behavior. Imagine sitting in a room filled with the soft hum of computers, each one processing thousands of viral genetic sequences, looking for patterns that might reveal the next pandemic threat before it fully emerges into our world. Machine learning algorithms now sift through vast databases of viral genomes, searching for the molecular signatures that distinguish dangerous variants from their more benign cousins. These digital detectives can spot concerning mutations weeks or months before traditional surveillance methods might notice them, providing precious time for preparation and response. It's like having a weather forecast for viral storms, though the predictions are still imperfect, and the storms themselves can change course without warning. Scientists have begun to map what they call the fitness landscape of viral evolution, charting the theoretical paths that viruses might take as they adapt to new hosts or environments. This landscape resembles a three-dimensional map of mountains and valleys, where each peak represents a particularly successful viral configuration, and each valley represents an evolutionary dead end. By understanding this terrain, researchers hope to predict which directions viral evolution is most likely to take. The concept of evolutionary forecasting has emerged from this work, attempting to anticipate viral changes before they occur. Much like meteorologists track weather patterns to predict tomorrow's rain, evolutionary biologists are learning to track genetic patterns to predict next season's viral variants. The mathematics are complex and the uncertainties are substantial, but early successes with influenza forecasting have shown the potential of this approach. Artificial intelligence systems are being trained to recognize the genetic signatures of increased transmissibility, immune evasion, and drug resistance in viral sequences. These systems can process information far faster than human researchers, scanning thousands of viral genomes in minutes to identify variants that warrant closer attention. Yet they still require human expertise to interpret their findings and make decisions about public health responses. The democratization of genetic sequencing technology has created a global network of viral surveillance that extends far beyond traditional public health institutions. University laboratories, private companies, and even citizen scientists can now contribute to our understanding of viral evolution in real time. This distributed approach to surveillance has both strengths and challenges, providing broader coverage while also creating new complexities in data quality and coordination. Real-time evolutionary analysis during outbreaks has become a powerful tool for understanding transmission dynamics and guiding response efforts. By tracking how viruses change as they spread through populations, scientists can identify transmission clusters, trace infection pathways, and monitor the emergence of variants with altered properties. This genetic epidemiology provides insights that traditional contact tracing and case investigation cannot achieve alone. The speed of modern viral detection and characterization has compressed the timeline for developing targeted interventions. Vaccines that once took years to develop can now be designed and manufactured in months, using platforms that can be rapidly adapted to new viral targets. Monoclonal antibody treatments can be selected and produced based on detailed knowledge of viral structure and function, providing precision tools for treatment and prevention. However, this acceleration of response capabilities has also highlighted new challenges in regulatory approval, manufacturing scale-up, and global distribution. Having the scientific knowledge to create effective interventions quickly is only the first step in a complex process that must balance speed with safety, innovation with equity, and individual protection with population-level benefits. The concept of pandemic preparedness has evolved to encompass not just the biological aspects of viral threats, but the entire ecosystem of factors that influence pandemic outcomes. Supply chain resilience, communication strategies, social support systems, and economic stabilization mechanisms are all now recognized as essential components of comprehensive preparedness plans. Scenario planning exercises have become increasingly sophisticated, using computer models to simulate how different types of pandemic viruses might spread under various intervention strategies. These simulations help policymakers understand the potential consequences of different response options and identify the most effective combinations of measures for different types of threats. The One Health approach to pandemic prevention recognizes that human, animal, and environmental health are interconnected in ways that require integrated surveillance and response systems. Monitoring viral evolution in wildlife populations, domestic animals, and environmental samples provides early warning signals about potential threats to human health, while also protecting the broader ecological systems on which we depend. Climate change is adding new variables to pandemic risk assessment, as shifting temperature and precipitation patterns alter the geographic ranges of animal hosts and viral vectors. Melting permafrost in Arctic regions may release ancient viruses that have been frozen for millennia, while changing weather patterns affect the breeding and migration patterns of animals that serve as viral reservoirs. The urbanization of human populations creates new transmission dynamics that must be factored into pandemic planning. Dense urban environments can facilitate rapid viral spread, but they also concentrate resources and expertise that can enable more effective responses. Understanding how viruses behave in different urban contexts helps inform both prevention and control strategies. International cooperation in pandemic preparedness faces ongoing challenges related to sovereignty, resource allocation, and information sharing. While viruses recognize no borders, human responses to viral threats are inevitably shaped by national interests, political considerations, and resource constraints. Building effective global cooperation requires addressing these fundamental tensions while maintaining the flexibility to respond to rapidly evolving threats. The role of non-governmental organizations, private companies, and civil society groups in pandemic preparedness has expanded significantly. These actors often have capabilities, resources, and connections that complement traditional government responses, but coordinating their efforts with official public health systems requires new models of partnership and collaboration. Technology platforms for pandemic response continue to evolve, from mobile applications that can facilitate contact tracing and symptom monitoring to blockchain systems that can ensure the integrity of vaccine supply chains. The effectiveness of these technological solutions depends heavily on public acceptance and participation, which in turn depends on trust, privacy protection, and demonstrated benefit. The psychological dimensions of pandemic preparedness involve helping populations develop realistic expectations about viral threats while maintaining appropriate levels of concern and engagement. This requires sophisticated communication strategies that can convey scientific uncertainty without undermining public confidence, acknowledge legitimate concerns while countering misinformation, and motivate protective behaviors without creating panic or despair. Educational initiatives aimed at improving public understanding of viral evolution and pandemic dynamics face the challenge of making complex scientific concepts accessible to diverse audiences with varying levels of scientific background. The goal is not to turn everyone into a virologist, but to develop sufficient understanding to support informed decision-making and appropriate responses to official recommendations. Healthcare system preparedness extends beyond surge capacity to include workforce training, supply chain management, and coordination between different levels of care. The COVID-19 pandemic revealed both the resilience and the vulnerabilities of healthcare systems worldwide, providing valuable lessons for future preparedness efforts. The economic aspects of pandemic preparedness require long-term thinking about investments that may not show immediate returns. Building and maintaining surveillance systems, stockpiling medical supplies, training specialized personnel, and conducting research all require sustained funding during periods when pandemic threats may seem distant and abstract. Legal and ethical frameworks for pandemic response continue to evolve as societies grapple with questions about individual rights, collective responsibilities, and the appropriate use of government authority during health emergencies. These frameworks must balance the need for effective public health action with protection of civil liberties and human rights. Research priorities for pandemic preparedness span a wide range of disciplines, from basic virology and immunology to social sciences and behavioral economics. Understanding how viruses evolve and spread requires not just biological knowledge, but insights into human behavior, social organization, economic systems, and political structures. The concept of Build Back Better has emerged as a guiding principle for post-pandemic recovery, emphasizing the importance of using the disruption caused by pandemics as opportunities to address underlying vulnerabilities and inequities. This approach recognizes that pandemic recovery is not just about returning to previous conditions, but about creating more resilient and equitable systems. Community resilience has become a central focus of pandemic preparedness efforts, recognizing that effective responses require not just top-down coordination, but also bottom-up engagement and local capacity building. Communities that have strong social networks, effective leadership, and adequate resources are better able to respond to and recover from pandemic disruptions. The role of traditional knowledge and indigenous practices in pandemic preparedness is gaining increased recognition. Indigenous communities often have sophisticated understanding of local ecosystems and animal behavior that can provide valuable insights into viral emergence and spread. Their traditional healing practices may also offer complementary approaches to prevention and treatment. Gender and social equity considerations have become more prominent in pandemic preparedness planning, as evidence has accumulated about the differential impacts of pandemics on different population groups. Women, racial and ethnic minorities, persons with disabilities, and other marginalized groups often experience disproportionate effects from both the direct health impacts of pandemics and the indirect effects of control measures. The environmental impacts of pandemic responses have emerged as an important consideration, from the increased use of single-use medical equipment and personal protective equipment to the reduced environmental monitoring and enforcement during emergency periods. Sustainable pandemic response strategies attempt to balance immediate health needs with longer-term environmental protection. Food security and agriculture systems have proven to be both vulnerable to pandemic disruptions and important factors in pandemic prevention. Changes in agricultural practices, wildlife management, and food distribution systems can all influence the risk of viral emergence and spread, while disruptions to food systems during pandemics can have cascading effects on public health and social stability. The mental health dimensions of pandemic preparedness have gained increased attention as the psychological impacts of isolation, uncertainty, and loss become better understood. Building mental health resilience and ensuring access to psychological support services are now recognized as essential components of comprehensive pandemic preparedness. Innovation ecosystems for pandemic preparedness involve not just traditional research institutions, but also startup companies, venture capital firms, and technology incubators that can rapidly develop and scale new solutions. These ecosystems require supportive regulatory environments, appropriate intellectual property frameworks, and mechanisms for translating research discoveries into practical applications. The role of citizen science in pandemic preparedness continues to expand, with opportunities for members of the public to contribute to viral surveillance, data collection, and community response efforts. These initiatives can extend the reach of professional surveillance systems while also building public engagement and understanding. As we settle into these contemplations of our collective preparations for future viral challenges, there's something both humbling and inspiring about the scope of human cooperation required. Across disciplines, institutions, and borders, countless individuals dedicate their careers to understanding and preparing for threats that may never materialize, working quietly in the background to protect populations from dangers they may never know existed. This vast enterprise of pandemic preparedness represents one of humanity's most ambitious attempts to learn from history while preparing for an uncertain future. It requires not just scientific knowledge and technological capability, but also wisdom about human nature, social organization, and the complex relationships between individual choices and collective outcomes. The viruses that may cause future pandemics are evolving even now, somewhere in the vast reservoir of animal hosts that share our planet. They are following the ancient rules of mutation and selection that have shaped life for billions of years, indifferent to human plans and preparations. Yet our growing understanding of these processes, combined with our expanding capabilities for detection and response, provides hope that future pandemics need not be as devastating as those of the past. In the quiet spaces between one pandemic and the next, this work continues. Scientists sequence viral genomes, public health officials update response plans, healthcare systems build capacity, and communities strengthen their resilience. It is patient, painstaking work that requires sustained commitment and investment over many years, often with little recognition or reward. But perhaps that is as it should be. The best pandemic preparedness is the kind that prevents pandemics from occurring in the first place, or minimizes their impact when they do occur. Success in this endeavor is measured not by dramatic interventions during crises, but by crises that never fully materialize, by outbreaks that remain contained, by communities that remain healthy and resilient in the face of viral threats. As we drift peacefully towards sleep, we can take comfort in knowing that around the world, this great enterprise of preparedness continues. Tomorrow will bring new viral sequences to analyze, new surveillance data to interpret, new strategies to test and refine. The work is never finished, but it is never abandoned either, sustained by the quiet determination of those who have dedicated their lives to protecting the health of populations they may never meet. The delicate balance between viral persistence and human adaptation reveals itself most clearly when we examine how certain viruses have learned to live quietly among us, neither disappearing entirely nor causing catastrophic outbreaks. These endemic viruses, as they're called, represent a kind of evolutionary détente, a negotiated peace between pathogen and host that allows both to coexist over the long term. Consider the common cold viruses that circulate through human populations year after year, causing sniffles and minor discomfort but rarely serious illness. These viruses have found their optimal strategy not in overwhelming their hosts, but in maintaining just enough presence to ensure their own survival, while avoiding the immune system's full wrath. They've evolved to be forgettable, in a sense, causing symptoms mild enough that we continue our daily activities, providing ample opportunities for transmission. This phenomenon of viral moderation isn't accidental, but represents millions of years of fine-tuning. Viruses that killed their hosts too quickly found themselves with nowhere to go, while those that cause no symptoms at all struggle to generate the coughs and sneezes that facilitate their spread. The sweet spot lies somewhere in between, creating just enough symptoms to aid transmission while keeping hosts mobile and social. The seasonal patterns we observe in many viral diseases reflect this delicate optimization. Influenza viruses time their annual appearances to coincide with weather conditions and social behaviors that maximize their transmission opportunities. The cooler, drier air of autumn and winter helps viral particles remain stable in the environment longer, while indoor gatherings and reduced ventilation bring potential hosts into closer proximity. But seasonality isn't just about environmental conditions. It also reflects the complex interplay between viral evolution and population immunity. As each flu season progresses, more people develop immunity to the circulating strains, making transmission increasingly difficult. The virus faces mounting pressure to evolve, to change just enough to slip past the immune defenses that worked against last year's variant. This annual cycle of adaptation and counter-adaptation has been playing out for centuries, creating the predictable rhythm of seasonal epidemics that we've come to expect. Public health officials use mathematical models to track these patterns, predicting when outbreaks will peak and how severe they're likely to become. These forecasts help hospitals prepare for increased patient loads and guide decisions about school closures and other interventions. Yet even within these predictable patterns, surprises can emerge. Sometimes a viral variant appears with mutations that confer unexpected advantages, leading to unusually severe seasons or outbreaks that occur outside the normal timing. These anomalies remind us that viral evolution, while following general principles, remains fundamentally unpredictable in its details. The concept of viral fitness landscapes helps us understand why some evolutionary paths are more likely than others. Imagine this landscape as a vast terrain of hills and valleys, where altitude represents the success of different viral configurations. Viruses naturally tend to climb toward fitness peaks, acquiring mutations that improve their survival and reproduction. But they can also get trapped on local peaks, unable to reach higher ground because the valley between requires temporary decreases in fitness. This landscape metaphor explains why viral evolution sometimes seems to stall or take unexpected turns. A virus might need to accumulate several mutations simultaneously to overcome a particular challenge, but each individual mutation might be slightly harmful on its own. Only when they occur together do they provide a net benefit, creating evolutionary leaps that can seem sudden and dramatic. The role of chance in viral evolution cannot be overstated. Mutations occur randomly, and whether a particular mutation spreads through a population depends on a complex interplay of biological properties and circumstantial factors. A highly beneficial mutation might disappear simply because the individual carrying it happened to be in the wrong place at the wrong time, while a neutral or even slightly harmful mutation might become common through sheer luck. This randomness is both a challenge and an opportunity for pandemic prediction. While we cannot forecast exactly which mutations will occur or when, we can identify the types of changes that would be most concerning if they did arise. Scientists maintain watch lists of potential pandemic threats, monitoring viral populations for signs of these worrisome evolutionary steps. The emergence of drug-resistant viral variants illustrates how human interventions can inadvertently shape viral evolution. Antiviral medications create strong selective pressure favoring variants that can survive treatment, much like antibiotics select for resistant bacteria. The challenge lies in using these powerful tools effectively while minimizing the risk of driving harmful evolutionary changes. Combination therapy represents one strategy for addressing this challenge, using multiple drugs with different mechanisms of action simultaneously. This approach makes it much more difficult for viruses to evolve resistance, as they would need to acquire multiple specific mutations at once. It's the same principle used successfully in treating HIV, where combination therapy has transformed a fatal disease into a manageable chronic condition for many patients. The development of resistance can sometimes reveal unexpected aspects of viral biology. When scientists first noticed that some influenza viruses were becoming resistant to a particular antiviral drug, they discovered that the resistant viruses were actually less fit in the absence of the drug. This finding suggested that widespread use of antivirals might actually select for weaker viruses, though this benefit could be temporary if the viruses later acquired compensatory mutations. Population genetics principles help explain why resistance emergence is so variable across different viral populations. In large, well-mixed populations, beneficial mutations spread rapidly and become common quickly. But in small, isolated populations, chance plays a much larger role, and beneficial mutations might be lost through genetic drift before they have a chance to demonstrate their advantage. The structure of viral populations within individual hosts adds another layer of complexity to evolutionary dynamics. During a typical infection, millions or billions of viral particles are produced, creating vast populations within which evolution can occur rapidly. Most of these variants are dead ends, but occasionally one emerges with properties that give it an advantage in spreading to new hosts. This within-host evolution can be particularly important in chronic infections, where viruses persist in the body for months or years. HIV provides a dramatic example of this phenomenon, with the virus continuously evolving within each infected individual to stay ahead of the immune response. By the time an HIV infection progresses to AIDS, the viral population may be genetically quite different from the strain that initially infected the person. The concept of viral quasispecies describes this cloud of related variants that exists within a single infected individual. Rather than thinking of viral infections as involving a single strain, scientists now recognize that most infections involve populations of closely related variants, each slightly different from the others. This diversity provides the raw material for rapid adaptation when conditions change. Bottlenecks in viral transmission create interesting evolutionary dynamics. When a virus spreads from one host to another, usually only a small subset of the total viral population makes the jump. This transmission bottleneck can eliminate much of the genetic diversity that accumulated during the previous infection, essentially forcing the viral population through an evolutionary reset with each transmission event. These bottlenecks help explain why some viral traits persist despite seeming disadvantageous. A mutation that reduces viral fitness within a host might still spread through a population if it happens to improve transmission between hosts. The virus essentially sacrifices some replication efficiency for better transmissibility, a trade-off that makes sense from an evolutionary perspective. The timing of viral shedding during the course of infection reflects another evolutionary optimization. Many viruses reach peak shedding levels before symptoms become severe, ensuring maximum transmission opportunities while hosts are still mobile and social. This timing isn't coincidental but represents millions of years of selection for variants that could spread effectively. Some viruses have evolved even more sophisticated strategies for optimizing their transmission timing. Certain respiratory viruses can manipulate host behavior, causing increased coughing and sneezing at precisely the times when viral shedding is highest. These manipulations are subtle but effective, enhancing viral spread without causing symptoms severe enough to keep hosts isolated. The phenomenon of viral interference describes how infection with one virus can protect against infection with related viruses, either through direct competition for cellular resources or through activation of immune responses that provide cross-protection. This interference can shape the ecology of viral communities, determining which viruses can coexist and which exclude each other. Understanding viral interference has practical implications for vaccine development and pandemic preparedness. If natural infections with mild viruses can provide protection against more dangerous related viruses, this knowledge might be leveraged for prevention strategies. Conversely, vaccination campaigns must consider how immune responses to vaccine viruses might affect susceptibility to wild viruses. The concept of original antigenic sin describes how early viral exposures can shape immune responses to related viruses encountered later in life. The immune system tends to rely heavily on memory responses from previous encounters, sometimes at the expense of generating optimal responses to new variants. This phenomenon can create complex patterns of population susceptibility that vary by age and exposure history. Maternal antibodies provide newborns with temporary protection against viruses that their mothers have encountered, but this protection comes with trade-offs. While maternal antibodies can prevent severe illness in very young infants, they can also interfere with the infant's own immune responses to vaccines, creating windows of vulnerability as maternal protection wanes, but active immunity hasn't yet developed. The role of viral evolution in vaccine effectiveness presents ongoing challenges for public health. Vaccines work by training the immune system to recognize specific viral features, but if viruses evolve to change those features, vaccine effectiveness can decline. This is why influenza vaccines must be updated annually, and why scientists are working to develop universal vaccines that target more conserved viral features. Next-generation vaccine platforms offer new possibilities for staying ahead of viral evolution. Instead of using whole viruses or viral proteins, some new vaccines deliver genetic instructions that allow the body's own cells to produce viral antigens. These platforms can be rapidly updated to match evolving viral sequences, potentially allowing vaccine updates to be developed and deployed much more quickly than with traditional approaches. The economics of vaccine development create interesting tensions with viral evolution. Developing vaccines is expensive and time-consuming, creating incentives to design vaccines that will remain effective for as long as possible. But viruses continue evolving regardless of human timelines and investment cycles, sometimes making expensive vaccine development efforts obsolete before they reach the market. Viral surveillance networks have become increasingly sophisticated at tracking evolutionary changes in real time. Scientists can now monitor viral populations globally, identifying concerning mutations within weeks of their emergence and assessing their potential impact on vaccine effectiveness or treatment options. This early warning capability is crucial for maintaining effective countermeasures against rapidly evolving threats. The democratization of genetic sequencing has transformed viral surveillance from a specialized activity conducted by a few major laboratories, to a distributed effort involving thousands of institutions worldwide. This expansion has dramatically improved our ability to track viral evolution, but it has also created new challenges in data standardization, quality control, and interpretation. Machine learning approaches are increasingly being applied to viral sequence data, searching for patterns that might predict future evolutionary trajectories or identify variants with concerning properties. These computational approaches can process vast amounts of sequence data much more quickly than human researchers, though they still require expert interpretation to translate findings into actionable public health guidance. The concept of viral fitness has evolved beyond simple measures of replication rate to include factors like transmissibility, immune evasion, and environmental stability. Modern fitness assessments consider the entire viral life cycle, from initial infection through transmission to new hosts, providing more accurate predictions of which variants are likely to succeed in different contexts. Experimental evolution studies allow scientists to observe viral adaptation in controlled laboratory settings, providing insights into evolutionary processes that would be difficult to study in natural populations. These experiments can reveal which mutations are most likely to emerge under different selective pressures and how quickly adaptation can occur under optimal conditions. The ethical dimensions of viral evolution research include questions about gain-of-function studies that might make viruses more dangerous, the sharing of sequence data that could potentially be misused, and the balance between scientific openness and security concerns. These issues require careful consideration of both the benefits of research and the potential risks of the knowledge generated. As we rest peacefully in contemplation of these intricate evolutionary processes, it's remarkable to consider that viral adaptation is happening continuously all around us, in every breath we take and every surface we touch. The microscopic world is in constant flux, with countless evolutionary experiments playing out simultaneously, most of them failures, but a few yielding innovations that will shape the future of life on Earth. This ongoing evolution is neither good nor evil from our human perspective, simply the inevitable consequence of imperfect replication in a changing environment. Viruses are not trying to harm us any more than rain is trying to spoil our picnics. They are simply following the fundamental imperative of all living things: to persist, to reproduce, and to adapt to whatever conditions they encounter. Our growing understanding of viral evolution provides us with unprecedented tools for anticipating and responding to future challenges, but it also reminds us of the fundamental uncertainty inherent in evolutionary processes. We can identify general principles and likely trends, but the specific details of future viral evolution remain as unpredictable as the weather, shaped by countless small events and chance encounters that cascade into large-scale changes. Perhaps there's a certain peace to be found in accepting this uncertainty while still working diligently to understand and prepare for whatever evolutionary surprises await us. The viruses will continue to evolve, as they have for billions of years, and we will continue to adapt alongside them, as our species has done throughout its existence. In this ancient dance between host and pathogen, each partner shapes the other, creating an ever-changing choreography of survival and adaptation that connects us to the deepest roots of life itself. The story of how we predict and prepare for the next pandemic is remarkably like tending a garden that spans the entire globe. Scientists scattered across continents work quietly, patiently, watching for the first signs of change in the viral landscape. They monitor the subtle shifts in genetic sequences, much like gardeners watch for the earliest hints of spring, knowing that small changes observed today might bloom into significant challenges tomorrow. In laboratories tucked away in university basements and government facilities, researchers maintain libraries of viral samples collected from around the world. These frozen archives represent snapshots of viral evolution captured at specific moments in time, creating a kind of genetic fossil record that scientists can study to understand how viruses have changed and adapted over decades. Each file tells a story of mutation and selection, of viral lineages that flourished or disappeared, of evolutionary experiments that succeeded or failed. The art of viral forecasting has grown increasingly sophisticated, borrowing techniques from meteorology, economics, and even astronomy. Just as weather forecasters track atmospheric conditions to predict tomorrow's rain, viral forecasters analyze genetic and epidemiological data to anticipate which viral variants might dominate future flu seasons. The mathematics are complex, involving probability distributions, phylogenetic analysis, and machine learning algorithms that can process thousands of viral sequences simultaneously. Yet for all our computational power and analytical sophistication, predicting viral evolution remains as much art as science. The researchers who excel at this work often describe it as developing an intuition for viral behavior, learning to recognize subtle patterns in sequence data that hint at functional changes. They speak of viral mutations the way wine connoisseurs discuss vintages, with a deep appreciation for nuance and complexity that comes only from years of careful observation. The global network of influenza surveillance represents one of humanity's most successful attempts at viral prediction. Every year, scientists around the world collect and analyze thousands of influenza samples, tracking how the virus is changing and making educated guesses about which strains will dominate the following season. This information guides the annual reformulation of flu vaccines, a process that must begin months before the actual flu season to allow time for manufacturing and distribution. The decisions made in those smoke-filled rooms where vaccine strains are selected carry enormous weight. Choose correctly, and millions of people receive protection against the coming season's dominant viruses. Choose poorly, and vaccine effectiveness plummets, leaving populations vulnerable to outbreaks that might have been prevented. The pressure is immense, yet the decisions must be made based on incomplete information and uncertain projections. Recent advances in viral surveillance have dramatically improved our ability to track emerging threats in real time. Genetic sequencing that once took weeks can now be completed in hours, allowing scientists to identify and characterize new viral variants almost as quickly as they emerge. This acceleration has compressed the timeline for pandemic response, creating new possibilities for rapid intervention, but also new pressures for quick decision-making based on limited data. The concept of viral fitness landscapes has revolutionized how we think about predicting viral evolution. Instead of viewing viral adaptation as a random walk through genetic space, scientists now understand that evolution follows predictable pathways shaped by the underlying biology of viral replication and transmission. Certain mutations are more likely to occur than others. Certain combinations of changes are more likely to succeed, and certain evolutionary outcomes are more probable given specific starting conditions. This landscape metaphor has practical implications for pandemic preparedness. By mapping the theoretical pathways that viruses might take as they adapt to new challenges, scientists can identify evolutionary bottlenecks where targeted interventions might be most effective. They can also anticipate which types of mutations would be most concerning if they did arise, allowing for more focused surveillance efforts. The role of animal reservoirs in pandemic prediction has become increasingly important as we've recognized that most pandemic viruses originate in non-human species. Monitoring viral evolution in wild birds, domestic poultry, pigs, and other animals provides early warning signals about potential threats to human health. Changes in viral genetics that might seem insignificant in animal populations could represent crucial steps toward pandemic potential. The challenge lies in interpreting these early warning signals correctly. Not every concerning mutation in an animal virus will lead to human infection, and not every virus that can infect humans will develop the ability to spread efficiently between people. The transition from animal pathogen to pandemic threat requires multiple evolutionary steps, each of which may be necessary but none of which is sufficient on its own. Climate change is adding new variables to pandemic prediction models, as shifting environmental conditions alter the geographic ranges and seasonal patterns of viral hosts and vectors. Warmer temperatures might extend the breeding seasons of mosquitoes that carry certain viruses, while changing precipitation patterns could affect the migration routes of birds that serve as viral reservoirs. These environmental changes create new opportunities for viral transmission and evolution that must be factored into prediction models. The urbanization of human populations has created novel environments for viral adaptation. Dense cities with extensive public transportation systems can facilitate rapid viral spread, but they also concentrate medical resources and expertise that can enable more effective responses. Understanding how viruses behave in different urban contexts helps inform both prediction models and response planning. Global trade and travel patterns have fundamentally altered the speed and scale of pandemic spread. A virus that emerges in a remote rural area can now reach major population centers within days through commercial airline networks. This connectivity means that local outbreaks can quickly become global challenges, but it also means that early detection in one location can provide warning time for distant populations. The psychological aspects of pandemic prediction present unique challenges for public health communication. Discussing potential future threats without creating undue alarm requires careful calibration of language and messaging. Scientists must convey the uncertainty inherent in prediction while still providing actionable guidance for preparedness efforts. The economic costs of pandemic preparedness must be weighed against the uncertain benefits of preventing events that may never occur. Investing in surveillance systems, stockpiling medical supplies, and maintaining surge capacity all require sustained funding during periods when pandemic threats seem distant and abstract. The challenge lies in maintaining political and public support for these investments over the long term. Research into universal vaccines represents one of the most promising approaches for staying ahead of viral evolution. Instead of targeting the rapidly changing surface proteins that most current vaccines focus on, universal vaccines attempt to train the immune system to recognize more conserved viral features that change less frequently over time. Success in this area could provide broader and more durable protection against diverse viral threats. The development of platform technologies for rapid vaccine and therapeutic development has accelerated dramatically in recent years. These platforms can be quickly adapted to target new viral threats, potentially allowing countermeasures to be developed and deployed within months rather than years. The COVID-19 pandemic demonstrated both the potential and the limitations of these rapid response capabilities. Regulatory frameworks for pandemic countermeasures continue to evolve as agencies balance the need for speed with requirements for safety and efficacy. Emergency use authorizations and other expedited approval pathways allow critical interventions to reach populations more quickly during crises, but they also require careful post-market surveillance to detect any unforeseen problems. International cooperation in pandemic preparedness faces ongoing challenges related to data sharing, resource allocation, and coordinated response planning. While viruses recognize no borders, human responses to viral threats are inevitably shaped by national interests and political considerations. Building effective global cooperation requires addressing these fundamental tensions while maintaining flexibility to respond to rapidly evolving situations. The role of private sector partnerships in pandemic preparedness has expanded significantly as governments recognize the importance of leveraging commercial capabilities and resources. Pharmaceutical companies, biotechnology firms, and technology companies all bring unique capabilities to pandemic response, but coordinating their efforts with public sector initiatives requires new models of collaboration and risk sharing. Community engagement has emerged as a critical component of effective pandemic prediction and preparedness. Local communities are often the first to detect unusual health patterns and the most important partners in implementing response measures. building trust and maintaining communication with diverse communities requires sustained effort and cultural competence. The democratization of scientific data and tools has created new opportunities for distributed pandemic surveillance and prediction. University researchers, private laboratories, and even citizen scientists can now contribute to our understanding of viral evolution and spread. This distributed approach provides broader coverage, but also creates new challenges in quality control and coordination. Educational initiatives aimed at building scientific literacy around viral threats and pandemic preparedness face the challenge of making complex concepts accessible to diverse audiences. The goal is not to turn everyone into an epidemiologist, but to develop sufficient understanding to support informed decision-making and appropriate responses to public health recommendations. The integration of artificial intelligence and machine learning into pandemic prediction systems continues to advance rapidly. These technologies can process vast amounts of data much more quickly than human researchers, identifying patterns and anomalies that might otherwise be missed. However, the effectiveness of these systems depends heavily on the quality of input data and the expertise required to interpret their outputs correctly. Social media and digital communication platforms have fundamentally altered the information environment during pandemic events. These platforms can facilitate rapid dissemination of public health information and enable new forms of community organizing and mutual aid. However, they can also amplify misinformation and conspiracy theories that undermine public health responses. The concept of pandemic phases has evolved to reflect our improved understanding of how viral threats emerge and develop. Instead of simple binary distinctions between pandemic and non-pandemic states, scientists now recognize a continuum of risk levels that can guide appropriate preparedness and response measures. This more nuanced approach allows for graduated responses that can be scaled up or down as threats evolve. Stockpiling strategies have become more sophisticated as we've learned from past pandemic experiences. Instead of simply maintaining fixed inventories of specific supplies, modern stockpiling approaches emphasize flexibility, surge manufacturing capacity, and just-in-time delivery systems that can rapidly scale up production when needed. The role of behavioral science in pandemic prediction and preparedness has gained increasing recognition as researchers have documented the crucial importance of human behavior in determining outbreak outcomes. Understanding how people are likely to respond to different types of threats and interventions is essential for designing effective preparedness strategies. Healthcare system resilience has become a central focus of pandemic preparedness efforts. Recognizing that the capacity to maintain essential services while accommodating surge demand is crucial for minimizing the overall impact of pandemic events. This resilience depends not just on physical infrastructure, but also on workforce training, supply chain management, and coordination mechanisms. The environmental dimensions of pandemic preparedness encompass both the ecological factors that influence viral emergence and the environmental impacts of response measures. Understanding how ecosystem disruption, biodiversity loss, and climate change affect pandemic risk is essential for developing comprehensive prevention strategies. As we settle into these contemplations of our collective efforts to anticipate and prepare for future viral challenges, there's something both humbling and inspiring about the scope of human ingenuity and cooperation involved. Across disciplines, institutions, and borders, countless individuals dedicate their careers to understanding patterns in data that may reveal tomorrow's threats, working patiently in laboratories and field stations, analyzing samples and sequences, building models and testing hypotheses. This vast enterprise of prediction and preparedness represents humanity's attempt to learn from the past while preparing for an uncertain future. It requires not just scientific knowledge and technical capability, but also wisdom about human nature, social organization, and the complex relationships between individual actions and collective outcomes. The viruses that may cause future pandemics are evolving even now, following the ancient rules of mutation and selection that have shaped life for billions of years. They are indifferent to human plans and preparations, pursuing their own evolutionary imperatives with neither malice nor mercy. Yet our growing understanding of these processes, combined with our expanding capabilities for detection and response, provides hope that future pandemics need not be as devastating as those of the past. In quiet laboratories around the world, scientists continue their patient work of viral surveillance and analysis. They sequence genomes, track evolutionary changes, build predictive models, and share their findings with colleagues across the globe. It is meticulous, painstaking work that requires sustained attention and careful interpretation, often with little recognition or reward. But perhaps that is as it should be. The best pandemic prediction and preparedness is the kind that prevents pandemics from occurring in the first place, or minimizes their impact when they do occur. Success in this endeavor is measured not by dramatic interventions during crises, but by crises that never fully materialize, by outbreaks that remain contained, by communities that remain healthy and resilient in the face of viral threats. The gentle rhythm of this ongoing vigilance provides a kind of comfort as we drift towards sleep. Somewhere in the world, a researcher is examining a viral sample under a microscope. A computer is analyzing genetic sequences. A public health official is updating response plans. The work continues through the night, sustained by the quiet dedication of those who have chosen to stand guard against invisible threats. Tomorrow will bring new data to analyze, new patterns to interpret, new predictions to test and refine. The work is never finished, but it is never abandoned either, carried forward by each new generation of scientists who inherit both the knowledge and the responsibility of protecting human health from viral threats. As we close our eyes and let consciousness fade into the peaceful darkness of sleep, we can rest knowing that this great enterprise of prediction and preparedness continues. The viruses may evolve and adapt with their ancient persistence, but so do we, armed with knowledge, tools, and an unwavering commitment to learning from the past while building a more resilient future. In the end, perhaps that is the most remarkable aspect of humanity's relationship with viruses. Despite countless challenges and setbacks, despite the inevitable uncertainties and occasional failures, we persist in our efforts to understand and prepare for these microscopic companions that have shared our evolutionary journey for so long. It is a testament to the human capacity for hope, cooperation, and quiet determination in the face of challenges that seem almost impossibly complex. And so, as the gentle science of viral evolution and pandemic dynamics settles softly into our dreams, we can find peace in knowing that tomorrow's guardians are already at their posts, watching and waiting, ready to sound the alarm if needed, but hoping always for the quiet success that comes when preparation meets opportunity, and when human wisdom proves once again to be our most powerful defense against the invisible forces that shape our world. The future of our relationship with viruses stretches out before us like an uncharted ocean, vast and full of possibilities we can barely imagine. As we lie here in the quiet darkness, drifting gently towards sleep, it's worth contemplating what the next chapters of this ancient story might hold. The tools we're developing, the knowledge we're accumulating, and the wisdom we're gaining from each pandemic experience are reshaping how we might coexist with these microscopic entities in the years to come. Imagine, if you will, a world where we can detect viral threats not weeks or months before they cause problems, but years in advance. Scientists are working toward creating what they call viral weather forecasts, systems that could predict pandemic risks with the same reliability that meteorologists predict storms. These forecasting systems would monitor the vast reservoir of viruses in wildlife populations, tracking their genetic changes and assessing their pandemic potential long before they make the leap to humans. The concept of viral weather might seem like science fiction, but the foundations are already being laid. Satellites now monitor environmental conditions that favor viral emergence, while networks of automated sensors sample air and water for genetic traces of novel pathogens. Machine learning algorithms sift through this constant stream of data, searching for patterns that human observers might miss, looking for the subtle signals that precede major viral evolutionary leaps. In laboratories around the world, scientists are learning to speak the language of viral genetics with increasing fluency. They can now predict which mutations are most likely to occur in specific viral lineages, and they're developing techniques to test these predictions in controlled environments. It's like having a conversation with evolution itself, asking what changes are possible and which ones would be most concerning if they occurred in nature. The dream of universal vaccines grows closer to reality with each passing year. Instead of chasing viruses as they evolve, these next-generation vaccines would provide broad protection against entire families of related pathogens. A universal coronavirus vaccine might protect against not just the virus that caused COVID-19, but also against future coronaviruses that haven't even emerged yet. A universal influenza vaccine could end the annual ritual of reformulating flu shots, providing decades of protection with a single series of immunizations. These universal vaccines work by targeting the parts of viruses that can't change without losing essential functions. It's like protecting against all possible car thefts by securing the engine rather than trying to protect against each individual key design. The engineering challenges are immense, but the potential benefits make the effort worthwhile. The speed of vaccine development continues to accelerate as new platform technologies mature. What once took years might soon be accomplished in weeks, with automated systems designing, testing, and manufacturing vaccines faster than viruses can evolve to evade them. The COVID-19 pandemic demonstrated what's possible when resources and attention are focused on vaccine development, and those lessons are being applied to create even faster response capabilities. But perhaps the most intriguing possibility on the horizon is the development of therapeutic viruses engineered to fight harmful pathogens on their own terms. These beneficial viruses could be designed to outcompete dangerous strains, occupying the same ecological niches, but without causing disease. It's a form of biological warfare fought at the molecular level, using evolution's own tools to protect human health. The concept of viral ecology is revolutionizing how we think about infectious disease prevention. Instead of viewing viruses as external invaders to be eliminated, scientists are beginning to understand them as integral components of complex biological communities. Managing viral threats might become more like ecosystem management, maintaining healthy balances rather than attempting total eradication. This ecological approach recognizes that removing one viral species from an environment often allows other, potentially more dangerous viruses to fill the vacant niche. The goal becomes shaping viral communities rather than eliminating them, encouraging the persistence of relatively harmless strains while preventing the emergence of dangerous ones. Personalized medicine is extending into the realm of viral threats, with the possibility of tailoring prevention and treatment strategies to individual genetic profiles and immune histories. Your personal viral risk assessment might consider not just your current health status, but your entire history of viral exposures, your genetic predispositions, and even the viral ecology of your local environment. The integration of artificial intelligence into viral research is producing insights that would have been impossible to achieve through traditional methods alone. AI systems can now design novel antiviral compounds, predict viral protein structures, and identify potential drug targets faster than human researchers working with conventional tools. These systems don't replace human expertise but amplify it, allowing scientists to explore possibilities that would otherwise remain hidden in the vast complexity of viral biology. Global cooperation in viral surveillance is evolving into something resembling a planetary immune system, with sensors and monitoring stations distributed across every continent and connected through real-time communication networks. This global sensing network could detect viral threats emerging anywhere on Earth and coordinate responses that transcend national boundaries and political divisions. The democratization of biotechnology is putting powerful tools for viral research into the hands of researchers around the world. Genetic sequencing equipment that once required major institutional investments can now fit on a desktop, while cloud-based computing resources make sophisticated analytical tools accessible to scientists regardless of their location or institutional resources. This democratization brings both opportunities and challenges. More researchers working on viral threats means more eyes watching for emerging problems and more minds working on solutions. But it also means that the tools for studying dangerous pathogens are becoming more widely available, requiring new approaches to ensuring that this knowledge is used responsibly. The role of citizen science in viral surveillance continues to expand as mobile technologies make it easier for ordinary people to contribute to scientific research. Smartphone apps can now collect and analyze health data from millions of users, creating vast datasets that can reveal viral spread patterns in real time. Participation in these systems is voluntary and privacy protected, but the collective intelligence they generate provides unprecedented insights into population-level health trends. Educational initiatives aimed at improving public understanding of viral science are becoming increasingly sophisticated and accessible. Interactive simulations allow students to explore viral evolution in virtual laboratories, while online courses make cutting-edge research accessible to curious minds around the world. Building scientific literacy about viral threats and pandemic dynamics helps create more informed and resilient communities. The economic models supporting pandemic preparedness are evolving to better capture the true costs and benefits of prevention versus response. Traditional economic analyses often underestimate the value of preventing low-probability, high-impact events like pandemics. New approaches are being developed to better account for the full social and economic costs of pandemic disruption, making the case for sustained investment in preparedness efforts. International legal frameworks for pandemic response continue to evolve as nations grapple with the balance between sovereignty and collective security in the face of viral threats. Treaties and agreements that establish protocols for information sharing, coordinated responses, and mutual assistance during pandemic events are becoming more sophisticated and comprehensive. The concept of viral refugees has emerged as climate change and environmental disruption force both human and viral populations to migrate to new territories. Understanding and managing these movements requires new approaches that consider the health implications of population displacement alongside traditional humanitarian concerns. Mental health support systems are being designed with pandemic resilience in mind, recognizing that the psychological impacts of infectious disease outbreaks can be as significant as their direct health effects. Building community resilience involves not just physical preparedness, but also emotional and social support networks that can help people cope with the stress and uncertainty of pandemic events. The integration of environmental monitoring into viral surveillance systems reflects growing recognition that ecosystem health and human health are intimately connected. Changes in forest cover, water quality, and biodiversity can all influence viral emergence risks, making environmental protection an essential component of pandemic prevention. Supply chain resilience has become a central focus of pandemic preparedness, as recent experiences have highlighted the vulnerabilities of global distribution networks. New approaches emphasize redundancy, local production capabilities, and rapid reconfiguration abilities that can maintain essential supplies during disruptions. The development of modular healthcare systems allows medical facilities to rapidly expand capacity during surge events while maintaining normal operations during routine periods. These systems use standardized components that can be quickly deployed and configured to meet changing needs, providing flexibility without sacrificing quality of care. Communication technologies designed specifically for pandemic response are being developed to ensure that accurate information can reach all populations, even when normal communication channels are disrupted. These systems prioritize reliability, accessibility, and multi-language support to ensure that critical health information reaches everyone who needs it. The preservation of traditional knowledge about infectious diseases and their management is becoming increasingly important as globalization threatens to erode indigenous and local health practices. Many traditional societies have developed sophisticated approaches to managing infectious disease risks that can complement modern scientific methods. Research into the social determinants of viral transmission is revealing new opportunities for intervention that go beyond traditional medical approaches. Addressing housing conditions, workplace safety, transportation systems, and social inequality can all contribute to reducing viral transmission risks in ways that are sustainable and equitable. The concept of viral justice recognizes that the impacts of infectious diseases are not distributed equally across populations, and that effective pandemic response requires addressing underlying inequities that make some communities more vulnerable than others. This approach emphasizes not just treating disease, but also addressing the social and economic conditions that contribute to its spread. As we drift peacefully towards sleep, contemplating these remarkable possibilities for humanity's future relationship with viruses, there's something deeply hopeful about the trajectory we're on. The tools we're developing, the knowledge we're accumulating, and the wisdom we're gaining suggest that future pandemics need not be as disruptive or devastating as those we've experienced in the past. The path forward isn't without challenges, and there will undoubtedly be setbacks and surprises along the way. Viruses will continue to evolve in unexpected directions, new threats will emerge from unforeseen sources, and our responses will sometimes fall short of our aspirations. But the overall trend is toward greater understanding, better preparation, and more effective cooperation in the face of shared challenges. The young scientists now beginning their careers will inherit tools and knowledge that would have seemed magical to previous generations of researchers. They will face viral challenges that we can't yet imagine, but they will also have capabilities that we can barely comprehend. The future of viral science belongs to them, and the evidence suggests they will use it wisely. In the quiet spaces between sleeping and waking, as consciousness fades gently into dreams, we can take comfort in knowing that this great human enterprise of understanding and preparing for viral challenges continues to evolve and improve. The viruses that share our world will continue their ancient dance of mutation and adaptation, but we will be there to meet them, armed with knowledge, tools, and an unshakable commitment to protecting the health and well-being of all people. And perhaps, in the end, that is the most beautiful aspect of this ancient story between humans and viruses. We are not passive victims in some cosmic struggle, nor are we all-powerful masters of our microbial fate. Instead, we are participants in an ongoing conversation that has been unfolding for millions of years, a dialogue written in the language of genetics and played out in the theatre of everyday life. Tonight, as you settle into the gentle embrace of sleep, remember that you carry within your own body the evidence of this remarkable partnership. Those ancient viral sequences woven into your DNA, those immune cells trained by countless encounters with microscopic visitors, that delicate ecosystem of microorganisms that calls you home. You are not separate from the viral world. You are part of it, shaped by it, and in your own small way, shaping it in return. The next time you catch a cold or receive a vaccination, take a moment to marvel at the intricate biological choreography unfolding within you. Your immune system, that masterpiece of evolutionary engineering, springs into action with responses honed over countless generations. The virus, following its own ancient imperatives, attempts to find its place in your cellular landscape. Neither good nor evil, Both are simply following the rules of life that have governed our planet since the very beginning. The scientists who work through the night in laboratories around the world, the public health officials who monitor disease patterns, the healthcare workers who tend to the sick, they are all part of this great human effort to understand and work with these invisible forces rather than simply against them. Their dedication represents something profound about our species, our capacity to learn, to adapt, and to care for one another in the face of challenges that seem impossibly complex. As your breathing deepens and your thoughts begin to soften at the edges, let yourself drift peacefully with the knowledge that this work continues. Somewhere, a computer is analyzing viral sequences, a researcher is peering into a microscope, a public health team is updating their response plans. The world is watching, learning, preparing, all while you rest safely in the quiet sanctuary of sleep. The viruses will continue their patient work of mutation and adaptation, as they have for billions of years and will for billions more. They will face the challenges we present to them, just as we will face the challenges they present to us. And in this ongoing dance of adaptation and counter-adaptation, both partners will continue to evolve, to change, to find new ways of coexisting in the remarkable experiment we call life on Earth. So sleep well, dear listener, knowing that you are part of something vast and ancient and beautiful. The gentle science of viral evolution surrounds you, supports you, and in ways both subtle and profound, makes you who you are. Rest in the knowledge that tomorrow will bring new discoveries, new understanding, and new reasons to marvel at the intricate web of relationships that connects all living things. And remember, wherever you are in the world tonight, whatever time zone cradles your rest, you are not alone. You are part of a global community of curious minds and caring hearts, all seeking to understand our place in this magnificent, mysterious, ever-changing world we share with countless other forms of life. Visible and invisible, simple and complex, familiar and yet still wonderfully full of surprises. Sweet dreams. And thank you for joining me on this gentle journey through the hidden world of viruses and the quiet science of pandemics. Until we meet again in another sleepy exploration, rest well, and may your dreams be filled with the peaceful wonder of discovery.